Let's look at ATP breakdown. ATP breakdown. Now, it's important to understand that ATP is only made to be broken down. It's kind of a weird concept, but this is the reality. It's kind of like that battery pack that I mentioned earlier, that additional battery charge that you have. You only charge it up to use it. Okay, you don't charge it up for it to be, held long term. It's temporary energy storage. So we only make ATP to then break it and use that energy to do cellular work. So in order to make ATP, we're going to need water. That makes this a hydrolysis reaction. Okay, so water comes in, it interacts with that ATP, and then it splits that third phosphate off, and we get ADP, that free phosphate again, and then some energy is now released. Remember I told you that between the bonds of that second phosphate and that third phosphate is a lot of energy. That's where we get the energy that we harness when we break this bond. Okay? And this is a hydrolysis reaction, as I mentioned. Okay, so with that said, that is how we break glucose down. Now we're going to start talking about the steps of cellular respiration, starting with glucose oxidation. Glucose oxidation. Now, when we talk about cellular respiration, we're going to speak specifically about what happens to one molecule of glucose, but I want you to bear in mind that we can break down other substrates. So we can break down fats. If we're on a ketogenic diet and we're avoiding carbs, the body will shift to breaking down fats as a source of energy. And in cases of extreme starvation, it will even start to break down proteins. Now, that's not ideal. Proteins are much more valuable in the body. They're needed in muscle, they're needed for hormones, they're needed for enzymes. So it's not ideal for the body to be breaking down proteins. But in the case of self-preservation, again, in starvation, we will start breaking down proteins. So bear in mind that 3 substrates can enter this process. But we're really going to focus on the main source, which is carbohydrates, which is glucose. Okay? Now, when you look at the reaction of energy metabolism for glucose, on the left here, this is a molecule of glucose, which has 6 carbons, hydrogen, oxygen. It's going to interact with oxygen. Oxygen is necessary for this process, right? And that is going to give us carbon dioxide, water, and energy. Carbon dioxide, water, and energy. Okay. Now I'm going to mention a lot of numbers here. You're not really expected to memorize those numbers in any way. They're just to give you a big picture and for you to understand what's taking place. You don't need too bothered with the numbers. So from glucose and oxygen, we then get carbon dioxide, water, and energy. And that is the first part of breaking down glucose to harness energy for cellular work. All right, remember, that's the big picture of what we're trying to do here. Now we get specifically 686 kilocalories per moles in this breakdown. And that is going to be a significant amount of energy, but we're in actuality not going to see or use all of that energy. We'll talk about that in a minute. So we're not going to use all of that 686 kilocalories per mole. We'll talk about that and why in a minute. I want you to bear in mind here that this reaction, although it does not have equilibrium arrows, it can occur in the forward direction as well as the reverse direction. Now, what is the process that we see this reaction occurring in the reverse direction? What's that process called? Where do we invest carbon dioxide and water and energy and make glucose and oxygen? What can take carbon dioxide and give us oxygen? What does that? Plants. Plants, perfect. So plants do that. Plants can take carbon dioxide, they can take water, they can harness some energy from the sun, and they can give us glucose, which is food, and they can also release oxygen, which we need. So that puts plants and human beings in a nice, harmonious, symbiotic relationship. Okay, they can give us oxygen, we can give them carbon dioxide. So what we give off, they need, and what they give off, we need, and so on and so forth. Okay. All right, so this is the reaction of glucose oxidation. We have glucose on the top with those oxygens, and then on the bottom we get carbon dioxide and water. Now the energy that is released off here, again, this process is not entirely 100% efficient. What does that mean? That means that all of the energy that we harness from breaking that glucose down is not going to be transferred to ATP. We're going to lose some of that energy in the form of heat. Okay, in the form of heat. And that just speaks to the law, the second law of thermodynamics, which is the law of entropy. There's a certain amount of disorder, a certain amount of randomness that happens in chemical reactions. So all of that energy that we harness, at 686 kilocalories per mole, all of it is not transferred to ATP. Instead, some of it is lost, a lot of it is lost in heat. Okay, and that's how our bodies generate heat. We have to lose heat as we undergo certain cellular processes. So the energy, whatever is harnessed, is going to be used to put that phosphate onto ADP. All right, so remember again, going back to that third phosphate group, putting that third phosphate onto ADP is charging up that batting pack. It's putting some energy away. It's storing up some energy in the form of ATP at the top. Okay. And now if we break that ATP again, we are going to release that energy to do some type of cellular work, to move molecules in the cell, to synthesize those molecules, to export them across the membrane. All of those types of activities require cellular work, and that's what we use ATP to do. We break it down, we take that energy, and we do cellular work. Okay. Now let's look at the reality of what happens here. So we have carbon dioxide in this equation in the box. We have carbon dioxide plus water plus ADP and phosphates. And we generate, we have glucose plus oxygen plus ADP and phosphates, and we generate carbon dioxide, water, and 38 ATP. Now this number can vary. It can vary based on age, it can vary based on sex, body status. It can vary one time in the day to another. So you'll see a lot of different numbers in different textbooks. Anywhere between 32 and I think 38. Okay, but we're going to use 38 today. So we make 38 ATP. 38 molecules of ATP is released from breaking down one molecule of glucose. So one molecule of glucose yields us 38 ATP molecules. Now, if we converted that ATP, those 38 ATPs to energy, we would only get 266 kilocalories. What does that tell us? What does that tell us? We broke down one molecule of glucose. We got 38 molecules of ATP, which is only going to give us 266 kilocalories per mole. On this reaction, there was 686 kilocalories per mole. Where did the rest go? Where did the rest of those, where did the rest of that energy go? I just mentioned it. To heat, perfect, to heat. So although we have the capacity, the potential in one molecule of glucose to make 686 kilocalories per mole of energy, in actuality, we only generate about 266 kilocalories. So 420 kilocalories is lost in heat, right? That's a lot, that's a significant chunk of the energy from that glucose being just evaporated into heat, okay? But that's just the way our bodies are, and that's just the law of thermodynamics. That's just what happens, okay? And so if we calculate the efficiency of oxidation, the efficiency of glucose oxidation, it's only going to be 38%, meaning of that 686, we only receive or get to use or get to harness 266 kilocalories. The remaining 61.2%, which is 420 kilocalories, is lost as body heat. So this process is not 100% efficient, but it's very, very useful, and it works. Okay, so let's get into the stages of glucose oxidation. The stages of glucose oxidation, there are 3 stages. Now, in between glycolysis and the Krebs cycle, there is a linker process. Right? There's a linker process in between there, and we'll talk about the linker process as we move on. Now, glycolysis and the Krebs cycle specifically are examples of substrate level phosphorylation. What did we say substrate level phosphorylation was? I compare that to something. Substrate level phosphorylation.
Stealing.
Stealing. Perfect. Thank you. So we're stealing a phosphate from some of the molecule. In this example, it's molecule X, and we're taking its phosphate, we're putting it onto ADP, and we're making ATP, and now that molecule goes off through without its phosphate. And examples of this are the glycolysis and the Krebs cycle. The Krebs cycle is also called the TCA cycle or the citric acid cycle. All three of those names are interchangeable. And finally, there's the electron transport chain, which uses oxidative phosphorylation. What did I say about oxidative phosphorylation? But you find a phosphate free floating. So ADP finds or stumbles upon a phosphate free floating, it attaches it to itself, and you make ATP that way. Okay?